So can everyone see my screen now?
Yeah.
Yes.
All right.
Okay, so good afternoon, everyone. Welcome to the Biophysics and Mechanical Biology Standardized Series. So today we have Professor Ming from MIT. He'll be giving a talk on biomechanical imaging of cells, extracellular matrix, and cell invasion in 3D. Dr. Ming is currently an associate professor in mechanical engineering and physics of living systems center at MIT, where he holds class 54 career development professorship. Before joining MIT in 2015, Ming obtained his PhD in applied physics and in 2014 and master's degree in 2012 in mechanical engineering at Harvard University and BS in engineering mechanics in Tsinghua University. For his research achievements, he received the Alpha Sloan Fellowship in Physics. So without further ado, Ming, the stage is yours.
All right. Thank you, Zhi, for the introduction. So first of all, I want to say it's the first or second day of Chinese New Year, the year of Ox. So I wish Happy New Year to everyone and a healthy New Year with a lot of strength as the Ox. So for today, I thought about it, what I want to do since we're really getting together to prepare for this center. So what I want to do is to go through a couple techniques and tools that my group developed in the last couple of years. For each of them, I'll show some applications, and hopefully this will help all of us to think about what potential topics and collaborations we may come up with. All right, so let me just get started. So what my group at MIT has been very interested in is to understand the mechanics of cells. We focus on single cells, extracellular matrix, and also when you combine them and when you combine many cells together, how do they construct a tissue with a particular functionality? And also during diseases, what happens in the crosstalk between cells and extracellular matrix. But in contrast, to think a lot more on the biochemistry side, what we have been very interested in is to think about cells as a physical fundamental building block of life. So when, for example, when you use different materials to construct a house, you want to have a certain parts to have a certain particular physical properties. Otherwise, it wouldn't have the final house with the function. So that's our focus. And of course, the long-term goal is with the fundamental understanding of the mechanics and physics of cells, we hope to diagnose disease, treat disease, and even prevent disease from happening in situ in the body. But it's certainly a long way that we're still rather at the first understanding step. So first of all, a little background of why we care about the mechanics of cells so much. Especially in my group, we focus quite a bit on the mechanics of cell interior. On the top left, you can see here's the fluorescent image of a cell that we stand a couple of different structures, actin, microtubules, lamentin, and also nucleus. The point is, it's not only very pretty, but you can actually see that the whole structure is entirely crowded. It's a highly jammed system. And next to it, you can see here's the TEM image that people use color to highlight some of the structures. I think red is actin, microtubule in blue here, the green is proteins. Again, the point is it's highly crowded. But if you simply just take a cell, put it under regular optical microscope, you will see enormous dynamics in the cell. Everything is constantly moving. Here are vesicles and the protein complexes. And on the bottom here is this the live actin. You can see the active filaments are also fluctuating. And you also see vesicles are being pumped away constantly. So the question is, in such a highly crowded environment, apparently, whenever you move, you will face obstacles. So the mechanical property of the surrounding environment plays a critical role in determining how you move. But how exactly all the movement come up, what energy is driving that is another topic that we study. In that environment, we have already showed in the last couple of years that if you directly influence the mechanics of the cytoplasm, for example, you compress the cell to concentrate it, or if you do mechanical perturbation to activate ion channels or something else, you start to change the mechanical environment of the cell. And that has a profound influence on many fundamental cell biology processes, for example, cell signaling, gene expression, and stem cell fate. One particular thing that I want to talk about is a signaling pathway called Wnt beta-catenin signaling, which is important in development, cancer, intestinal system. So it's a very important fundamental cell signaling pathway. And what we find is if you concentrate the cell interior materials by taking water out or using other perturbations to have a more crowded interior, you can actually increase the concentration of the proteins that is required for this pathway. And that will increase the signaling activity a couple times higher. And using this method, we can actually engineer intestinal organoid by compressing it. We make it grow actually even faster and having more stem cells. So if you're interested, you can take a look at this paper. But I won't spend too much time talking about this today, but rather I want to focus on the tools that we developed. All right, so since the focus of our work is the mechanics of cells, we have a major way that most of the people in my lab will use is optical tweezers. What we typically do is take a cell, you either grow it on 2D or you embed it in 3D. And then what we do is we take some polystyrene particles, half micron or even a micron large, and we either let the cells endocytose them in the cell or use a glass needle to directly inject them into the cell. And then what we do is we use a focused laser beam to trap a particle. And then, just like my hand, I use the laser beam to grab the particle and then push it in one direction at a constant speed. And this will apply a force on this particle, and using this particle and basically deforming the surrounding cytoplasm. And that will give us a direct force displacement response curve that we use to characterize the mechanics of the cytoplasm. So on the video here, if you take a look, one particle on the right in the cell is being dragged to the right, to the edge of the cell. And correspondingly, the force displacement curve is highlighted here inside. That is fluctuating, but roughly you can measure the spring constant or calculate the stiffness if you wish. So first of all, I want to say that the mechanics of the cells and mechanics of the cytoplasm is highly complex, is not a trivial material. Some of the early work by Jack Fredberg shows the power law of rheology that we see it repeatedly. And there are many other people sometimes also claim that it may be a viscous fluid, and some recent papers even claim it's a paroelastic material, that when you deform the cytoplasm too quickly, you push water around it first. Here we saw all of those in the same cell by using a probe to deform the cytoplasm at different speed. So depending on how you load your system, how quick, how slow, how large your probe is, you can actually see a very diverse types of responses in the living cell. But this is only a result in the linear regime that we simply just deform the cell a little bit. And in my lab, what we have been more interested in the last 2 or 3 years have been on the nonlinear mechanics, where that you deform the cells a lot. You stretch the cells too much, higher than 50%, even 100% strain, or you take the particle to drag the particle in the cytoplasm to a long distance. What happens? We care about this because and when the cells are in their physiological condition, they constantly migrate and deform. So all of those are associated with large deformation. That's why we become quite interested in this. And the other reason that we are very interested in large deformation mechanics is because of a particular cytoskeletal component called intermediate filament. The type of intermediate filament we play with is called Biomechanical. It's the type of cytoskeletal intermediate filament that you can find in mesenchymal cells. Classical behavior, such as epithelial to mesenchymal transition, you will see cells start to express momentum, which arguably will allow the cells to migrate and deform. Those intermediate filaments are known to be responsible to many, many different diseases. I think 70 or 80, many of which are related to laminin, but also to vimentin and keratin that we know about. But it's still unclear what exact role that these intermediate filaments play in the cell, especially the cytoskeletal ones. So why cells have them is really unclear. Some of the hypotheses have been assuming that they're important for the mechanics of the cells, that we actually show it here in this work. So to study the mechanics of cells with and without intermediate filament, or in particular how intermediate filament contributes, we collaborated with Bob Goldman's lab at Northwestern University. We transfected the cells to have a pair that one with Biomechanical intermediate filament, the other without, is completely knocked out. I think Bob compared actin and microtubules stay the same. It's a constant for a number of passages. So using these, we can directly probe the mechanics of the cell when they have intermediate filament or when they do not have intermediate filaments. You can imagine we can start to put particles in and drag them to measure how stiff they are. But at this point, I was still not very happy about it, because even you have a cell pair with Biomechanical, the other without, it's still not a pure Biomechanical. You still have a lot of other things in the cells. So it's very hard to compare, I still believe. So we actually created a somewhat interesting system that we call it Biomechanical intermediate filament ghost cell, or enriched cytoskeleton. So the way we do it is taking advantage of the property that intermediate filament have a much slower turnover rate. They don't constantly depolymerize and depolymerize. It's much slower. Because of this property, we basically use some chemicals to wash away the cell membrane, depolymerize actin, wash away microtubules, and it gave a few gentle wash to get rid of most of the proteins. And then what's left over is this. It looks still like a very beautiful cell, but without any fixation. So we didn't fix it. All you see, the fibers here, are actually only Biomechanical intermediate filaments. So it's just a ghost that you see maintain their natural shape and morphology of intermediate filaments. However, they don't really have anything else. For this system, we can basically study the mechanics of the pure Biomechanical intermediate filament network. And the way we do it is, again, very simple. We just put particles in before we start this preparation, and then we go through all the steps. And in the end, we'll have a network of Biomechanical and with particles trapped inside, which allow us to, again, use our optical trap to start to play and measure it. All right, so some quick results is that when we start to drag particles in these cells, first, what I showed here is the wild type cells with Wymentin, and the green is the same cell type, but without Wymentin. And you can see that when I do the dragging in the wild type cells, the force increase initially because you're deforming more and more materials as you move further. At some point, when the strain is roughly 100%, you start to yield the network. But in the cells without Wymentin, you actually see that happens much earlier. Only half, 50% of the strain you start to yield the cell, and the strength, which is the peak stress that the cell can take, is also a lot smaller. The interesting thing is this: when we do our measurement in the GHOST system, where we only have Wymentin intermediate filaments, you can see that initially the force displacement curve is quite low. They're really floppy, those networks. So they're not contributing to too much resistance initially. But when you drag more and more, they start to take over. So at the large deformation where the wild type yield, it does seem like the majority or the main resistance is actually from intermediate filaments because they take over the strength and stretchability at the end. So the other thing I didn't really include, but what we can also do with the optical tweezers is we can grab a particle, drag them in one direction, and then we can unload. So you can actually do cyclic loading that material scientists usually study material. And with that, we can measure energy dissipation and toughness of material, for example. And in this case, we actually find the cells with Wymentin intermediate filament has the higher toughness, is almost the 3, sometimes I think even 4 times higher than the cells without Wymentin intermediate filament. So the conclusion here is cytoskeletal intermediate filament seems to be a very important player for cell behavior at large deformation. You need them when you deform a lot.
Yep. Ming, can I ask a question?
Sure.
Yeah. So this graph makes it look like, to a kind of a crude approximation, that the control cell, the wild type, is the sum of just the intermediate filament contribution and everything else. But yeah, I'm sure that there are interactions between intermediate filaments and other components. But this, to a 0th order, it looks like it's just additive. Is that overly simplistic?
No, well, is a little simplistic. It does seem like it's just some, but exactly as you said, there are physical interactions, which is actually very important. Let me just show you this model, then you see what I'm talking about. So here is what we think is going on in the cytoskeleton. When you only have F-actin and microtubules, you have a local loading. The force, when it's large enough, it basically would yield the surrounding material. It either depolarize or something, then your force doesn't go out. So stress and strain doesn't propagate out. But when you have intermediate filaments, they can take a lot higher force. And therefore, when you have the same local loading, the stress and the strain can actually go out, be transduced to a larger area because you have intermediate filaments. And by doing that, they actually involve more actin and microtubules into any local deformation. Therefore, that's the maximum that we think the toughness is being increased. But this also highlights the physical interaction that perhaps because Wymentin exists, they actually interplay with the rest of the cytoskeleton, and this physical interaction may actually enlarge the space that where a local loading is impacting. But I think a 0 order assumption is fair. This physical interaction seems to play some role as well.
I have a question. Yeah. So I want to go back to the basic a little bit here. So when you say intermediate filament here, so you mostly are you substituting Wymentin with all the intermediate filament? What about other things like lambing and all the stuff? Or am I getting that confused? I thought lambing is
This is an excellent point. This is all the work are for Wymentin, but I believe things like keratin is the same thing. You probably have seen a paper by Shalvia tripod. It was published on Nature, I think 2 years ago, about how keratin is contributing to hyperelasticity or superelasticity of a cell monolayer. That's roughly the same idea, I think. But lamin, I don't know, since they're only surrounding the nucleus, and there are different subtypes. So I don't know for sure how this idea may play the role there.
So here's you're not, so the measurement that you have, because it's away from the nucleus, so you think that not the nuclear mechanics doesn't really matter.
Yeah.
So, no. Okay. So it's waiting to talk about the intermediate filament that are away from the nucleus, so to speak.
Yeah, exactly. But thank you for the good point. So since I'm really just talking about the tools, I should say all the measurement now is away from the cell cortex and away from the nucleus. But the tweezer, our tweezer is strong enough that if you want to measure the cortex or the nucleus, we can do that. But for this particular study, it's not. All right. So another thing that we can do on our setup is while we have a laser beam to grab a particle, we can actually image the surrounding cell structure to see how the deformation is being propagated. And this confirmed the conceptual picture I just presented. On the top row, this is the cell with intermediate filament, 1-19 intermediate filament, and the white circle is the particle. And what we do here is we fluorescently label some of the cellular structure and then do a PIV to measure the displacement field. You can see that when particles move, the displacement field propagates into a larger range. And in the bottom row, this is a cell without 1-19, and now you can see the same displacement of the particle, but the whole deformation field is much smaller. Okay. And you can calculate the strain field, you see the same thing. So if in some of your systems, you want to apply some local loading while looking at how much deformation you're inducing, this tool will be perfect for that, I think. All right, so just one quick thing about this. Why do we care and how do we know this actually matters, the existence of 1-19? And another thing that we do in the lab is we embedded the cells that either have or do not have 1-19 into a bulk hydrogel, and then we start to stretch the hydrogel and force the cell to be compressed and stretched. Basically deform, I think in this case, a 300% strain, and we maintain that for half an hour to an hour. And you can see that if you do the same thing for the cells with 1-19, most of them actually can be alive, being stretched for half an hour. But if you get rid of 1-19 in the mass, they die very quickly as long as you stretch beyond 100% strain. So I think that highlights the important role of 1-19 intermediate filament in maintaining cell mechanical integrity under large deformation per se. That may be why they need it for EMT, which we haven't really tested, but that's I think a reasonable hypothesis. Jing, do you have another question?
Yeah. So how do you embed the hydrogen cells in the hydrogel without killing it?
This is a collaboration with Xuanhe Zhao at MIT. This is their tact gel. I think interpenetrated with something. Since typical hydrogel, also you can't stretch them 3 times longer. This is their double network gel and modified the polymer to have cell adhesion site. But still, you can't just culture the cells in there forever. This is really, quick experiment for a couple hours. Cells seem to be okay.
I see. Yeah. Thank you.
I think longer than 12 hours, they may actually have trouble. All right. That's the first part. I basically wants to show our typical methodologies to measure mechanics of cells. So far on individual cells, but we can actually apply all those tools to measure multiple cells. For example, when you have a cluster in 3D, that if time allows, I'll talk about that as briefly as well. So since the living systems is not only composed of cells, they have enormous space filled with extracellular matrix. We also are very interested in the mechanics of the extracellular matrix, especially how cells interact with the extracellular space and how they feel this interaction. Jeff, you have a question?
No.
Okay. All right. So the motivation is very simple and clear. We know that when you grow cells on substrate of different stiffnesses, the cells can sense it and feel it. That's typically done when you grow cells on a 2D hydrogel whose stiffness is fixed, that you know was the stiffness that you're dealing with. But cells in reality are often in biopolymer matrix in 3D, which are, for example, collagen, fibrin, and those materials. And they are often nonlinear and they're dynamic, that cells can change. For example, here is we grow one human breast cancer cell in a 3-dimensional collagen gel. You can see as the blue cells spread out into a spindle shape, they actually collect the collagen fibers near the two spindles. And that changed the alignment, that changed the concentration. And the question we ask is, does that change the mechanical properties also? And going back to the knowledge that we know cells can sense the mechanics of their surrounding environment, if they change their surrounding mechanics, do they sense that? That's the question that we still don't know the answer, but I'll show you some of the early results that we have. The question is, how do cells change the mechanics in their surrounding matrix? And the method is very simple. We simply, again, just put particles into the collagen while we polymerize the sample in cells, embed the cells. In the end, we have a collagen gel with the cells spreading in it, and we have particles here and there at different locations. And the particles are large enough that they're trapped by the collagen mesh. Therefore, we allow us to use the, again, optical tweezer to trap them and grab them to measure the force displacement curve, which we again use to measure the mechanics, the stiffness per se. All right, just some very quick results. When we measure at different distances to the cell, for example, if there's a particle really far away from a cell, and you grab the particle to measure their local stiffness, what you get is somewhere here. So this is the far away stiffness, and you can convert it to compare with the shear modulus. For example, you measure with bulk rheology, and that's consistent. And the interesting thing we find is when we move closer and closer to the cell, you actually see a drastic stiffening behavior. The closer you are to the cell, the stiffer the matrix are. And when you are really close to the cell, you see the matrix stiffness, in this case, collagen can stiffen by 2 orders of magnitude. In some special cases, we even see that the stiffness of the collagen can increase by 3 orders of magnitude. And one thing that we initially thought about is maybe because cells are collecting collagen, they're densifying the collagen surrounding them, therefore the increased collagen density can explain this. So we did some experiment and also some numerical estimation. For example, based on the measured collagen concentration, you can estimate how stiff they should be. You don't get a nearly 2 to 3 orders magnitude stiffening. And in fact, when we stop cell contraction in this case, the cells are not contracting anymore, and then the stiffness increase actually dropped down to the line where roughly you can still estimate based on the concentration or density of the fibers nearby. So this huge stiffening in the collagen is actually by the cell contraction. And why this is happening, it's actually because not only collagen, it's been shown that most of the biopolymer matrix are nonlinear. What this means is when you start to deform a biopolymer matrix, a little bit is okay, but the more you deform them, the stiffer they are. the explanations are is not in agreement. There are different ways to explain it, but that's a very common belief. So what we think is going on is as the cells spread out in the 3D collagen, they generate the contractile forces, and that contractile forces will induce the contractile stress field that is the largest near the cell. Therefore, that contractile stress will induce the local collagen matrix into a very strong nonlinear stiffening regime. And because the contractile stress decay over distance, you see that basically slowly disappear as we move away from the cell. So it's really the strongest near the cell because the contraction is strong. So the stiffening is not due to concentration, but it rather is a result of cell contraction induced the nonlinear stiffening.
Ming, just a question, like when you have these beats close to the cells and so on, like does it matter, like, if you, like push them along the direction of the fibers or toggle the fibers, you would probably measure like a different stiffness, right?
Yeah,
so
that's an excellent point, Andrew. So I should have said, all the measurement I show here are along the cell spreading direction. If I measure perpendicular direction, you still have some stiffening, but not as large. Yeah, that sort of confirmed the idea of contraction also, I think, because the contraction is along this direction. All right. So since we know that the local matrix stiffening is a result of the contractile stresses induced by cell contraction, one question we have been wondering about is whether we can actually measure or infer the local stress from the measured matrix stiffness, because the stiffness is the result of the stress. Can we go backward to actually know how large the stress is a very important thing that to measure contractile stresses has been at the beginning been done on 2D. That method developed by Jeff and others, that you put cells on 2D hydrogel with tweezers or use pillars, as Chris Chen did in his lab, and then you grow cells on it. When cells start to contract, you know the substrate property so well that you can measure the strain, and using the property, you can calculate the stress. And this has been shown to be a useful, very useful indicator of disease or different status of the cell. So it's something that we actually really want to know. But as we know, cells are also often in 3D environment, in biopolymer environment. So people in the field have been trying to think about a way to measure the traction stresses in 3D. It's been done by a couple groups using different sort of a method similar to the 2D traction force microscopy. For example, Chris Chan's group and Guy Genning's group, they embed cells in a linear elastic hydrogel, which again, you know the elasticity, so you can measure the strain field based on tweezers, and then you can measure the stress, you can calculate stress field. And there are other people, Ben Fabry and Vivek Choi, what they have been doing is they assume a constitutive relation for a biopolymer matrix, collagen or some others. And they again embed tweezers to measure the strain field, and then using the constitutive relation to calculate the stress field. But it's a very lengthy process that also require a case-by-case computation power, and you also need to relax the cell in 3D, fully relaxed, to get a reference that allow you to measure the strain. So none of those are trivial. So we have been thinking, based on our local mechanics measurement, can we get at least a proxy of the stress without going through any of those steps? And the answer is yes. It's actually a quite simple method. We know that collagen and many other biopolymers are nonlinear, right? That means the higher stress you apply onto the gel, the stiffer they are. Okay. And for example, here's the results from the waste lab show actin gel has the same behavior. When you put the gel under a balchriometer, you start to apply stress. The larger stress you apply, the stiffer they are. So the y-axis is the differential stiffness, x-axis is the pre-stress. At very small pre-stress, when the material is in linear regime, the stiffness doesn't change. But as long as you're in the nonlinear regime, there's a monotonic increase that a certain stress will give you a certain stiffness. And that makes us wonder whether we can just simply go backward. Since we know the stiffness, can we just read out the stress that corresponds to a certain stiffness? It sounds very simple, but it requires some modeling to actually firmly demonstrate. And we also did a numerical modeling as well. I'm not showing any of those, just going through the fundamental idea quickly here. Since we measured a nonlinear stiffness reference curve of the collagen by doing a local pulling away from a cell, that we know as we apply larger and larger force, the matrix becomes stiffer and stiffer. And we know that near the cell, the matrix is very stiff. And as we move away from the cell, they become weaker and weaker. So we very simply just take the measured stiffness and using this reference curve to read out the corresponding stress in any of the measured location. And that allows us to directly measure the stress any point that we have a particle. We can do that. So one thing I want to highlight a little bit is typically I would say we hate nonlinearity because nonlinearity is giving us so much trouble in calculation, measurement, everything. But here in this method, we call it nonlinear stress inference microscopy. We actually love nonlinearity, since if you're in linear regime, a large stress variation wouldn't change the stiffness too much. But in the highly nonlinear regime, a little bit stress change will change the modulus a lot. Therefore, you can actually have a very precise measurement by doing this backward reading. So if you're interested in the theoretical details, you can read more details in this paper. So to summarize this part, we can use this to measure local matrix stresses in different types of biopolymer matrices when we have different cells in it. Collagen, fiber, and matrigel are the three major ones that we measured. For these different types, collagen and fiber are strongly nonlinear, and matrigel is actually weakly nonlinear. They have a very large linear regime, but as long as you have some nonlinear regime, you can use this method. Right, so here I basically talked about our effort in measuring the mechanics of the ECM in situ, that we can directly measure that in space and in time as the system develops or so. And we can even use this to backward calculate the stress. So if any of you are interested in this, I think there were some interesting things that we can try. All right, any questions about this part?
Yeah, Ming, I have a question here. So you show this for this material, which is basically strain stiffening. Does this also apply to strain softening or even yielding material where you have a curve that is more going downwards?
So first, I think yielding likely will not work because it's not monotonic anymore. Softening, I think so. I don't see why not. Yeah.
Okay.
Softening, I think so. Yielding most of
it has to be reversible. It has to be
reversible as well. Yeah. It has to be elastic actually.
Mm-hmm. I've got a question, Ming. So that you're saying you point out here that it's a -2 power law with distance. I'm wondering physically what that means. If we're linear material with a point force, you might expect 1/r. But is this because it's like a forced dipole?
Yeah, no.
Is that where the square comes from?
That's an excellent point. I got rid of 3 slides for explaining that because I don't think I have enough time. But that's exactly it. So we're plotting here the stress, right? If you have a point of force in a 3D linear material, you need an area to times the stress to balance the force. So the stress should decay quadratically. But a cell is a forced dipole, right? So that will induce tension and compression. They actually cancel each other. So in Londos and the Lifshitz book, there's a explicit solution saying for forced dipole in 3D, it should decay 1 over the distance cubed. So it really shouldn't be quadratic.
Oh.
So it should be faster. But remember, all the materials that we're dealing with are not homogeneous materials, they're fibrous materials. And what it means is when you stretch them, they're okay, but when you have compression, they buckle. And if a fiber buckle, the compression doesn't propagate any further. So as a result, in force dipole induced force field, you only have tension in your system. You don't have any compression that goes. And that goes back to a point force case, is that you only have quadratic decay. It's actually very, deep physics in there, I think. But it also has very interesting implications that perhaps when a cell contracts strong enough, they can stiffen their surrounding. And this stiffening, as a result of stress, decays slower because the stress decays slower. So they actually influence a bigger field of space that perhaps they can use this to talk to each other. That's something that we are currently quite interested in.
And what does this say about if you have multiple cells in the same matrix? What does this say about kind of the shielding distance, about how far away does the cell have to be before it feels its neighbor?
So for example, if you look at this here, I think we have the stress decay, for example, across 20 microns where you still have a Pascal or something. So it probably will increase a radius of 20 micron or so. But only for this large force. If you have, if there's a tumor cluster, for example, I think the field of view is even larger.
Right.
I'm Leo. I have a question. So regarding the figure A issue there, right? So, and my understanding, whatever you measured, I think are probably reflecting two things. One is pre-stress, the other one is a local modulus of the gel itself. And when you're using a curve like A, it's kind of, at least you think that the local modulus changes of a gel doesn't matter. The other way to say it is, your curve on the figure A, how does that apply to region very close to cells, and how does that differ from, which is, let's say, 20 micron away from the cells?
You mean the effective? Yeah, I think that's actually the question we're asking ourselves now. So if you have a linear elastic materials, the deformation or something doesn't matter exactly because they don't change anything, right? You think you put cells on a material of a kilopascal, that's it. But here in collagen, you think you're putting them in a collagen that has 20 Pascal stiffness, but it's not. It's maybe 200 or 2000 Pascal. So it has a huge effect, perhaps. That's another thing that we're trying to do, whether this nonlinear stiffness behave as a linear stiffness, that have the same impact on, for example, James pressure or something. But I believe so.
Hey, Ming, I have a question about your comment on the stretching versus buckling.
Yeah.
So were you saying that there's no buckling observed?
There is. We actually went back to the gel near the cell. We saw tons of buckling. And we did an experiment and we focused on one piece of the gel as a cell migrating toward this direction, being buckled into the tongue.
Okay. Yeah, that's cool. And do you see it buckles into a multi-wiggle mode?
Yeah, that we didn't really define, but yes, it's it's pretty weird a shape because the geometry of the network is complex. We have some images in this paper. You can take a look.
Yeah, that's great. Is it buckled in kind of 2D fashion or 3D? Do you know?
I can't really say. We see a lot of 2D ones, but we also see part of them out of plane, I think. But it's hard to say.
Okay.
Yeah, and defining buckle for collagen is hard anyway because when they naturally polymerize, they're not super straight, right? They have some curvature already. So it's not something simple, I think.
Okay, thanks.
All right, so let me just very quickly talk about one more thing. Is our we're actually become in the last 2 years, I would say we are very interested in multicellular systems now. Not only single cells or their ECM, but when you have a piece of tissue growing in your lab, how they interact with the surroundings. For example, what I show here is a model system pioneered by Dave Mooney, Joanne Brucke, and Nina Bissell. That you can see if you put a single breast epithelial cell, MCF10A, in a piece of hydrogel that is soft, they basically maintain cellular shape, just like a breast SNR structure. But if you put them into a stiff matrix, they can invade and become very much like a malignant cancer. And the question we have been asking is, what about the mechanics of cells during this process? How much do they change themselves? How much do they change their surrounding matrix? How do they change the way that they talk to each other? So it's a long process. We started by probing the mechanics of the cell and cell migration, per se. Let me just show you something quick. So by the way, this model system that we have been using recently is everything starts from an MCF10A cell in a 3D alginate plus matrigel matrix. And they just divide and form this cluster, and after day 5 or 6, they start to invade. At the end of 10 days to 2 weeks period, they behave very much like aggressive malignant cancer. And if you compare that to what we see in a human carcinoma sample, you see a very similar geometry. And here's the normal one. And one more interesting thing before I go in is, arguably EMT has been seen in many of the cancer events. In this particular model, we actually see that as well. In the invasive tip, you start to see more Wyminton, a little less ECAD, but staining everything in a 3D cluster is very hard, so we can't really say for sure, but there's some evidence, I think. All right, the first thing we did with this model system is we scan fluorescent cell nuclei that allow us to see each cell, and then you can track them, you can measure their size, you can measure their shape. This is how they look like at the 5th day, that the whole cluster is spinning as a whole. But if you put the cluster back to the incubator for another 5 days, this is how they look. So you start to see that they're sending out the invasive branches, the cells go out and come back in, they're trying to do something really hard. I'll show results of tracking the cells, but I want to start with the mechanical measurement.
Can I ask you a question, Ming?
Yeah, sure.
Why do you call this breast cancer? These are MCF10A cells.
Well, there are a few different reasons. I want to say this is a model cancer spheroid, since they're from breast, so I call them model breast cancer. And we see some markers, and we have a base membrane at the beginning. When this starts to invade, we see some of the signaling. I think that we compare with what I forgot who it was, but some of the PS3K signal at the invasive tips, you also see that. So you actually see reasonably amount of evidence that is consistent with a mammary cancer type.
Right, but MCF10A cells are not cancerous cells. These are taken to be normal cells. And what you're showing looks more like branching, what would be considered to be branching morphogenesis than to be what you're calling metastatic behavior.
Yeah, no, that's absolutely right. So I don't think we can call that metastasis or malignant cancer. But for example, if you try to look at Wyminton, you do see an upregulation at the tip of the branch.
But Vimentin upregulation in mammary epithelial cells happens normally during morphogenesis. It's not the expression, like you can't infer that expression of Vimentin means that a breast cell is acting like a cancer cell. I guess the objection I have to your slide here is that you're calling MCF10A cells a breast cancer spheroid, and the breast cancer community is going to say absolutely not.
Yeah, I actually didn't know the upregulation of Vimentin is normal. I thought they're a marker of EMT. That's why I think.
Well, sure, but you can have EMT take place during normal development, right? That's how EMT was discovered. It's a normal developmental process that is actually quite controversial in its activation during tumor progression.
Yeah. Okay,
I mean,
that's good to know.
If you want to be looking at cancer, then you should use cancer cell types, not normal cells.
Yeah, so initially we actually have been struggling with how to call that for a long time. So thank you for pointing this out. Yeah, so for a MCF10A cluster, when they have invasive tips and the tips starts to express some of the surface markers, for example, and what else do you think one should compare if you want to further compare them with breast cancer?
I wouldn't compare them with breast cancer.
Yeah.
I think that's a false equivalence.
Okay, that's good to know. Yeah, I didn't think too much about that because we borrowed this model from Dave Mooney's group, and then they look very much like breast cancer. Then we start to play with them to call them microtumors, but then I didn't feel comfortable with calling them tumors, and then I sort of say maybe it's okay to call them, 3D breast cancer model. Yeah, I should go back to check, some of our other evidence to confirm this. All right. So let me just continue to show what do we do. So since we really want to measure the mechanics of each individual cell in the system, the way we do it is just as how we measure the cells on 2D. We put particles in it, and then we shine a laser to grab the particle and to drag them in the cell to measure the mechanics of each of the cells.
Yeah. I'm sorry, can I interrupt? Can we go back to Celeste's question? I just want to finish that off. So Celeste, would it be okay to just call this a model of invasion and just take away breast cancer? I
would call this a model of collective migration or collective invasion, but you certainly cannot call normal cells cancer cells,
right.
Because they have a phenotype that looks like a section of a tumor.
Right.
At least not for the cancer biology community.
Right. So we can just call it what it is.
Mm-hmm.
Was an invasion.
Invasion is okay, just not cancer. I see.
Yeah. Just
it's the one word there that raises objections.
So you'd be okay with that
on this for a long time.
Yeah. Okay. Good. Thanks, Celeste.
Thank you. All right. So let me just very quickly show you what we do. By using the laser trap, we measure basically each cell within this cluster. Just show you this result on the right. So at day 3, when the cluster only has a few number of cells, they roughly have a similar stiffness. None of them are too stiff, none of them are too soft. But when when the cluster starts to grow, you have some cells stiffer, some cells softer. And at the 10th day, when they start to invade into surrounding matrix, some cells are really stiff and some cells are really soft. And the interesting thing is the stiff cells are all the cells in the core, and the softer ones are the ones in the branch. So these invading cells become much softer. Another thing that we typically do, since we have particles inside, is basically by looking at how particles will fluctuate. And from that, we can infer the dynamics within a single cell. Another thing that we developed with a few others, based on the fluctuation and the mechanics, is we can directly measure the force that induce the spontaneous fluctuation of the particles or organelles in a single cell. That using this method, we can directly measure the force. Approximately, you can think this as the aggregate force fluctuations or enzymatic activity within a cell. That we also see that those invasive cells are the most active ones, and the cells in the core are the least active ones. The cells in the branch are constantly trying to do something. They have a stronger force. They're also softer. Here is to track their migration. That you certainly see those cells migrate faster, and the cells in the core migrate a lot slower. There's a very clear difference. But you actually also see that within this cluster, cells constantly migrating out, and they also migrate back in. While they do that, they actually change all of the physical properties that we have measured dynamically. When they go out, they become softer, they speed up. And another thing that I haven't shown yet is they swell themselves, they become larger. And the reason that they swell themselves is because they have more water and dilute everything inside, therefore they reduce the stiffness. So the point of this part is, since we ran out of time, is we're using optical tweezers so we can measure the mechanics of each individual cell throughout their development in space and in time. At least in this a model system that is less than 500 cells. When you have too many cells, we may do it selectively, but for a smaller evolving system, we can see every detail very clearly. I think I will just stop here. I don't think I have time to talk about everything. So just stop at the conclusion that you can go back to some of the work that we published. All right, so thank you all for your time and the suggestions. That's very valuable.
Okay, thanks, Ming. Yeah, it's been an hour, but if you have to leave, yeah, that's okay. But if you have more questions, I think Ming can probably stay for a few more minutes.
Well, I have a few questions about your sphere. So first of all, do we know why they have to reach a certain size before they become invasive? And also, secondly, how soft is the? So you have a core that is the most rigid, and then you have the peripheral being softer.
Yep.
How the matrix, is the matrix softer than the cell at the peripheral? Is there such a...
Yeah, that's an excellent point. So the matrix also stiffen, very similar to the single cell case. When there's the invasive branch, they actually stiffen the surrounding matrix also. And the matrix, I think, is actually softer than the cells. All of the cell stiffness we measure is in the cytoplasm, but if you measure the cell cortex, it's different. And the matrix, the collagen matrix or alginate matrix in this case, is only a couple hundred Pascal. Cell cortex can be a kilopascal or so. So they're slightly stiffer than matrix.
So how about the first question?
Oh yeah, sorry. The first question. They actually often starts to invade at the 4th or 5th day when they have a reasonable size. I don't know why they have to wait until then to invade. But early on, they also try to establish basement membrane and everything to protect themselves and have some, additional layer that they can interact with. That may be why, but I don't really know.
Or did people look at whether maybe certain genes start to be activated? Maybe it takes some time for some EMT factors to get expressed.
So very recently, there's a paper, I think by O.V. Charter's lab, starts to look at a gene expression in those systems, and it starts to see some changes, but I don't know if they really follow every day.
Okay.
What people have done is compare the end point and the beginning point, I think. But over the time, you can see some of the signaling pathways starts to be upregulated. That could contribute as well.
Ming, I have a question here. So you see this spheroid, so I missed the numbers there. Are they much stiffer than the matrix gel on the outside, right?
They are, well, the outside is alginate plus matrix gel. In this case, it's 300 Pascal shear modulus.
And
so they are stiffer, but they're not much stiffer, I think.
They're comparable, so to speak.
Yeah. Stiffer, okay. but on the same order.
Okay, and then the other question is that in the spiral case, so you see the differentiation in terms of the stiffness. How much of that is due to stiffness sensing, and how much that is due to differentiation, and how much is that just due to nutrient limitation in the middle of the spheroid?
Well, this is actually something interesting. So the cells in the middle of the spheroid can move out, and the one outside can move in. So at this small size, I don't think, and because they're so dynamic, I don't think the nutrient limitation would be a huge factor in this case. But I think the pressure being built up may be a factor. That's how we explain the physical parameters.
So because the core, basically the core is in the is under higher pressure, so they respond to that and they develop a stiffer phenotype.
Right.
Okay, cool. Thanks.
So I mean, if we look at the shape of the this spheroid after invasion happens, I wonder what factors would determine that shape. Like, does the initial shape of the spheroid matter, or maybe the distribution? I mean, the stiffness of the surrounding matrix.
My guess is the heterogeneity in the matrix that actually triggered this. But you can see the cell population is also very heterogeneous. Some of them may interact with the surrounding matrix very strongly, and that will induce a bigger difference in that local matrix, right? So I think that feedback is inducing their invasion, and that changed the morphology also.
So if we, let's say, embed the spheroid in a bilayer matrix, right at the interface, do you expect the shape may look different from when you just embed it in a homogeneous?
I think so. I think if you have a very heterogeneous matrix, you will start to see difference immediately. You may see things invading in one way or compared to the other.
Have you tried that? I also wonder whether something interesting might happen at the interface.
No, we haven't. That is actually a very interesting idea. No, we haven't.
Okay. Yeah, maybe let's try. Great.
Hi, I have a very quick question.
Sure.
Okay, so just wondering, like when you tested the intracellular mechanical responses, do you find those mechanical responses actually homogeneous or somewhat heterogeneous?
Is actually heterogeneous depending on where you probe. So what we do is we just measure a bunch and then take average. But that's right. It's not homogeneous at all.
Okay, I see. And also, is it isotropic or anisotropic?
It depends on whether the cell shape is isotropic or anisotropic. If the cells spread out in one direction, then the mechanics is very anisotropic.
Okay, I see.
Yeah.
Thanks.
Great. Any last we can take one last question if there's any.
Ming, I have a further question regarding your spheroid. Do you see, you always see those pretty nice isotropic shape. Can you sometimes see like this anisotropic shape? Like in total, the spheroid, a spheroid became like more like elliptical or oblate?
We haven't, but I know if you make your gel, for example, has a gradient of stiffness, and then you will see some elliptical shape. But if the gel is homogeneous, I don't think you see that.
What if you just increase the stiffness of the environment of the gel?
I mean, we haven't done that, but I think the Moody's group did some. They see that it starts to irregularly grow immediately. You don't seem to have an ellipsoid state.
Okay, that's interesting. Thank you for. Yeah, so at some point I can show some of the we do a very different thing. We do microbiology, but we have some similar observations. We will come to ourselves, so I can talk at some point to see, and I'm always looking for some similarity in eukaryotic systems. So we'll talk to you later.
Okay, yeah, that sounds really cool.
Okay. Well, thanks. So yeah, thanks, Ming, again.
All right, thank you.
All right. Happy New Year to everyone.
Bye, everyone.
Yeah.
Bye-bye.
Bye.
Bye.
Thanks.